Hi everyone, and welcome to today's NHLBI Small Biz Hangout. I'm Gary Robinson, the Business Development Advisor in the Office of Translational Alliances and Coordination here at the NHLBI, and today's presenter will be Dr. Chris Sassiella, Regulatory Specialist in our office. And she'll be presenting on the topic of integrating your regulatory and research plans for biomedical product development programs. So just a brief disclaimer to let everybody know that we are presenting these Hangouts for educational purposes. They're not meant to be guidance for a particular product or technology. If you're developing something that falls within the scope of the NHLBI mission, please contact us through our website given here for additional guidance. If you're working outside the scope of our mission, we encourage you to reach out to an appropriate program official in the institute, NIH Institute, relevant to your disease area or organ system, and you can learn more about that at the NHI SBIR website, sbir.nih.gov. The slides are available if you email us at either of the following email addresses, NHLBI_SBIR@mail.nih.gov, or you can email Chris directly. She'd be glad to provide you with copies of these slides. We will also be archiving this event and captioning it, and it will be on our NHLBI YouTube channel some point within the next month or so. So look out for that. And again, sign up for our listserv, gave that URL earlier to get updates on new events and funding opportunities. And with that, thank you very much. Thank you, Chris. A few housekeeping matters. If you're having problems joining the video session, you can try to leave and rejoin the event. In terms of hearing the audio, if you're not hearing me, well, I guess you're not going to hear my advice, but otherwise, if you're looking at the slides, you can stream through your computer. Click the Call Me icon on your screen, enter your phone number, or simply call in using the toll-free number here and the event number here below that. And that call-in number and event number will be at the bottom of the subsequent slides. And I want to let you all know that today's event is being recorded for archiving and future listening and viewing. You'll have the opportunity to ask questions, and Chris will answer them along the way during the course of the presentation. Only we will be able to see your questions, you can submit questions at any time during the presentation using the chat function on your screen. If you're in full screen mode, you would need to hover over the chat icon on your screen, and a pop-up will come up where you can enter your questions. Sometimes people will submit similar questions on the same topic, and if this happens, we'll address them with a single answer. So your specific question may not get answered. You can, as I said, enter questions anytime during the event, and due to the general nature of this presentation, questions specific to your particular technology won't be addressed. The NHLBI Small Biz Hangouts are intended to provide general information about topics that may be unfamiliar to many biomedical researchers who are just starting out as entrepreneurs. The information conveyed here is not product specific and shouldn't be taken as a complete approach for any specific technology. The advice provided in these events may be useful to guide innovators in putting together product and business development plans for their companies. This series includes sessions on regulatory matters relevant to different technologies, understanding market and customers for a product, developing and defending intellectual property strategies, and points to consider relating to getting reimbursement coverage for a new technology. All of these Small Biz Hangouts are recorded, captioned, and posted to the NHLBI YouTube channel, and the URL for that is given here. We announce upcoming Hangouts and other educational opportunities as well as funding opportunities with periodic emails to a listserv. Please sign up for that email listserv here. using the following URL, bit.ly/NHLBI capital -SBIR capital updates. The cases are actually important when you're using a bit.ly URL. So do a little bit about our office. I just talked briefly about our educational opportunities. These include the Small Biz Hangouts and guest lectures. The main function of our office is to coordinate the small business program for the National Heart, Lung, and Blood Institute. So in that process, we provide advisory services on business development and regulatory affairs to prospective applicants and awardees. We have entrepreneurs in residence and investors in residence with significant experience in the biotech world. We also connect entrepreneurs and applicants with our NHLBI program officials who are the domain area experts for the therapeutic indications that you might be studying. And we also connect you and make you aware of any guidance coming out of the NIH Central Office of the Director relating to SBIR policies and rules. In addition, in our office, we run two translational research programs, the NIH Centers for Accelerated Innovations and the Research Evaluation and Commercialization Hubs, which are directed at advancing early-stage technologies from universities into companies. So our presenter today is Dr. Chris Sassiella, who's the regulatory specialist, as I mentioned, in our office. Chris has 20 years of research experience, which includes development and optimization of blood and plasma therapeutics and a deep background in bioassay and screening platform development. She also has 9 years of regulatory experience, which started with a fellowship at the US FDA, where she learned regulations and how product reviewers think about new technologies as they're being developed. After her fellowship, she joined a contracting company where, as the head of the regulatory affairs and services team, she was responsible for supporting multiple additional aspects of developing, executing, and maintaining the full spectrum of regulatory communications and documentation for multiple clients. Chris joined the NHLBI 3 years ago and has been working with our biomedical innovators to help them understand how to identify and comply with U.S. regulatory development requirements for their new products and technologies. She received her PhD in pharmaceutical sciences from the University of Maryland School of Pharmacy and is a certified professional through the Regulatory Affairs Professional Society. Today, Chris will describe how to perform some basic regulatory research, using that information to guide a technology development plan and integrating the research and regulatory plans together when writing an SBIR or STTR application. And so with that, I would like to pass the presentation over to Chris.
Today, we're going to focus on understanding how to develop a preliminary regulatory strategy, use that information to ensure that you're doing the right experiments to progress your product development efforts, and how to convey that information in your small business application to NIH to give the reviewers confidence in your ability to really bring that product to the commercial market. So what do I mean when I title this presentation, Integrating Your Regulatory and Research Plans? So an integrated approach to product development is sometimes called a critical path, or a streamlined development, or a product development plan. Basically, what this indicates is that the experiments or other work that you're performing is going to be used to support market access for your technology. So some of the questions that you should be considering from the very beginning stages of your technology development effort are in there, right? What is the expected regulatory pathway? What are the uncertainties or challenges that you can foresee for your technology? And how are you going to proactively address those as you continue to pursue your technology development? And this is something that you really should consider regardless of whether or not you plan to bring your product to market or if you plan to license it or sell it to another party. You're going to need to be able to explain clearly and logically how far you've progressed your technology and how much more work needs to be done. So in these Hangouts, we like to use case studies, and our hypothetical case study today is going to be a Phase 2 SBIR application for an ultrasound imaging technology. The intended use for this ultrasound will be to image blood flow in the peripheral vasculature. Since we're looking at an SBIR Phase 2 application, there's a 12-page research plan, and there's also a 12-page commercialization plan. Information from both of these sides of the application should be reflected in the research plan. So here's a sort of very vague set of specific aims that might come in with a Phase 2 application for this technology. Specific aim 1, develop a prototype new device. Specific aim 2, perform bench testing on that device. Specific aim 3, perform some clinical studies on that device. And then specific aim 4, refine the device based upon the first 3 specific aims and get some sort of okay from the FDA in order to get to market. So this particular application might not really go over spectacularly because it seems a little bit vague, but it is our starting point. Let's take a look also at a commercialization plan that might come along with this. Again, another 12 pages. You would talk about how peripheral artery disease is an unmet need with anticipated growth because of our aging population. You might talk about limited device competition in this space or some nuance of your technology that will enhance readouts compared to current technologies. You talk about how solid your intellectual property protection is around the novelty of your technology. But oftentimes, the regulatory plan will read as simply as, we're going to license or sell the technology prior to FDA submission. So really, we don't need to know a whole lot about that regulatory stuff. Or we'll submit a 510K application, but with very few or potentially no details about what's required for that application. So one thing I want to do before we move on too far is I want to call out some of the other Hangouts that we've already developed and that are available on our YouTube channel. And one is one that Gary delivered last year around this time called Writing Your Phase 2 Commercialization Plan. It's a much more extensive discussion about how to perform market research, convey the medical need for your technology, and how you're going to identify customers. And it's a great resource. So I encourage you to take a little bit of time and take a look at that when you have, a spare moment or two. So in terms of developing a reasonable regulatory plan for the development of your technology, first you're going to need to understand the FDA, how it works, where within it your technology is going to be regulated, and what kind of public information you have access to while developing your regulatory approach before you actually talk to the agency. Biomedical product review at FDA happens primarily in 3 centers: the Center for Biologics Evaluation and Research, the Center for Drug Evaluation and Research, and the Center for Devices and Radiologic Health. Because the case study I have chosen today is an ultrasound device, and the Center for Drugs and the Center for Biologics have similar development pathways, what I'm going to do today is I am not going to talk about the Center for Biologics. I'm going to focus in on these other 2 centers to describe how FDA works. So let's talk about the Center for Drug Evaluation and Research. Within CDER, new drug review happens in offices, and there are 4 offices of drug evaluation, ODE 1, 2, 3, and 4. There's also an Office of Antimicrobial Products, an Office of Hematology and Oncology Products. So since Office of Drug Evaluation isn't really very descriptive about what gets reviewed in those offices, let's take a little bit deeper look at what they review. Each of them contains 3 divisions, and those divisions are listed here on this slide. I'm not going to read them all to you. So I want you to just take a note, though, that ODE 4 contains medical imaging products. So if our product contained a new imaging agent in addition to an ultrasound technology itself, that might fall under the purview and be regulated by this division in ODE 4. So how do you know if you're developing a drug? A drug is something where the primary intended use of the technology is achieved through chemical action or being metabolized by the body. a biologic is something that is biologic in origin. There's a little bit of fuzziness because some biologics are actually regulated by the Center for Drugs. But generally speaking, if the product acts on the body in a chemical or metabolic way, then it's going to be likely a drug or a biologic. And if you're developing a new drug or a biologic, your general development pathway in the U.S. is going to be to perform preclinical testing, and that testing will need to be sufficient to demonstrate reasonable probability of safety for first-in-human studies and development of a reproducible and scalable manufacturing method so that if you are approved, you can supply your drug to the public. You're going to provide that initial preclinical information to the FDA in a document called an Investigational New Drug Application, along with your first clinical protocol and a bunch of other documentation. The FDA will review that and either allow you to proceed or put you on hold. If you're allowed to proceed, you will then conduct a series of clinical trials to demonstrate first the safety of your technology and to identify a potential dose range, and then to expand in exposure to larger groups, including those with the indication you're targeting, and to look for indicators of efficacy while continuing to accumulate safety information. And then finally, of course, you'll need to conduct pivotal trials while continuing to accumulate safety information in larger groups, comparing your technology to the standard of care and confirm its effectiveness. If all of these studies are successful, you may end up filing either a biologic license application, a new drug application, or an abbreviated new drug application as appropriate to your technology to request a commercial marketing for your new therapeutic. Chances are, if you're listening to this webinar, you're at the very early stages of this process, and it may be indeed your very first time engaging in a drug development project. So you may be wondering where you can go to find information about how this process actually works. So the FDA, because of the Freedom of Information Act, posts much of the data that supports the preclinical testing of therapies online. The databases listed on this slide contain a lot of information about how new drugs were developed and what additional information was submitted with each label claim expansion. There's also information in the Orange Book about patents, and in the inactive ingredient database about molecules generally considered to be non-therapeutic that you may end up including in your final formulation for your technology. For a fuller understanding of how to effectively search these databases and the types of information you can glean from the publicly posted documents, I encourage you to check out our prior Hangout, Developing a Target Product Profile. So we've covered sort of drug and biologic regulatory research. Let's move on to medical devices. In the Center for Devices and Radiologic Health, the review of new technologies is performed in review panels, and review panels are organized into two offices, the Office of In Vitro Diagnostics and Radiologic Health and the Office of Device Evaluation. So if you take a look at the Office of In Vitro Diagnostics and Radiologic Health, or OIR, review panels, you'll see down at the bottom of this slide that there are potentially two panels where a ultrasound technology might be reviewed. Potentially, if it were for a breast indication, it could go into the mammography quality standards section, so that's less likely, or it could go into the radiologic health group. So for right now, let's think that this is where our case study may be regulated. Now let's take a look at the Office of Device Evaluation. Within this group, the review panels are, as you can see, they're right here. The ultrasound technology that we're going to be using as our case study today, if we were looking to see where it might be regulated, we would think about our intended use for the technology, which is to look at the peripheral vasculature. And the review panel that would best fit that indication or that intended use is the cardiovascular review panel. So now we've identified two potential review panels for our technology, and we're not quite sure, but we're at least sort of on a road. Let's take a brief review of how medical devices are regulated here in the U.S. at the FDA. A medical device is defined as a technology that does not achieve its primary intended use through chemical action or by being metabolized. So basically, that would be something that is mechanically separated from the body or is not integrated at a molecular, cellular, tissue, or whatnot level. Medical devices are regulated by the FDA based upon the potential risk of the technology, and they're stratified into 3 classes. Class 1 are very low-risk medical devices and are essentially subject to minimal oversight. Class 2 devices are considered moderate-risk technologies, and they are similar to existing technologies, and they may be able to prove what is called substantial equivalent to a prior existing technology called a predicate. Class 3 medical devices are high risk, and here I want you to think about life-sustaining types of technologies or technologies that might guide a clinical decision in a life-sustaining or life-threatening situation. Class 3 medical devices are subject to a development path that's much more closely aligned to that of drugs and biologics, with a requirement for clinical proof of safety and effectiveness. Class 1 technologies, which are either very low risk or extremely well understood, may be exempt from most regulatory oversight, often needing only to file some paperwork with FDA prior to entering the open market. Class 2 devices, or moderate risk devices, typically enter the market using what's called the 510K pathway. And this is where a manufacturer will demonstrate the substantial equivalence of their new technology to a prior technology. So you may have a single predicate, which is your primary basis for substantial equivalence, but you're also bringing in some novel aspects in your technology that you're pulling from other secondary technologies. That is also acceptable. Some, but not all, 510K eligible technologies will require clinical studies, and some, but not all of those studies, those clinical studies, may need to occur with FDA approval, which would be garnered by filing an investigational device exemption, which is a bunch of documentation similar to that you provide for an IND, but more along the lines of biocompatibility and structural and design element testing. The highest risk, or Class 3 technologies, require clinical proof of safety and efficacy conducted under an IDE prior to the submission of a pre-market approval application. So this is a much larger burden of regulatory review compared to a 510K, which is a much smaller set of data. Now, you'll notice in the Class 3 section here, I also have de novo 510K, and I just told you that Class 3 is high risk. But sometimes Class 3 really is just something that is brand new, that has never existed before, and for which there is nothing to claim substantial equivalent to. So for devices that are so technologically novel that may be manufactured using 3D printing, and there weren't any prior approved technologies using a 3D printer, a manufacturer can apply for market access using what is called the de novo 510K process, which has a lower burden of regulatory documentation than a pre-market approval, but slightly higher than a more standard 510K. So how do you find out what sorts of documentation you might need to submit for a Class 2 or a Class 1, Class 2, or Class 3 technology? For Class 1, it's a pretty standard filing, so we're going to jump right into Class 2. If you want to do some research on your Class 2 device, there is very nicely a searchable database on the FDA website for the 510K pre-market notifications. And you can use any single field on this search box. So you can, if you happen to know the product code, for instance, or you know the name of the currently marketed device and you want to put in a device name, or you know what panel reviews the type of technology that you're interested in developing, you can do a search of one or multiple combinations of these fields, and what you will get returned is a list of what are called 510K summary statements. And by reviewing those 510K summary statements, you can begin to understand the types of testing that the manufacturer had to perform in order to achieve their market access. If you are developing a Class 3 device, there's a similar database that you can search for pre-market approvals. And again, you can search using any single or multiple fields. And what the result will be here, what will be returned to you here, is a much fuller set of review documentation. So there's a lot more meat that you can sink your teeth into, but even given that wonderful benefit, it's usually hoped by developers that they are a 510K Class 2 device versus a PMA Class 3 device, because the development pathways are quite a bit longer for PMA devices. Not everything is a Class 2 or a Class 3 PMA all by itself. As I mentioned earlier, there are de novo technologies, or there may be technologies that have humanitarian device exemptions, those that have very small markets. There are a lot of different types of technologies, and so there are additional research databases on the Center for Device and Radiologic Health website that you can take a look at and really use these to develop a strong understanding of the regulatory path for your technology. There is also one more place I'd really like everyone to take a look before they engage in in-depth product development work, and that is the FDA guidance database. About a year and a half ago, FDA very kindly took all of their guidance documents and put them into a single database. Prior to that, you had to go searching through each center, and it was a little bit more cumbersome. It's much easier now. It's so easy that you can go in and type in even just a single word, such as ultrasound, and out of the 3,339 total entries that were in the database on the day that I performed this search, there were 5 that popped up. So it really is an extremely useful and powerful search engine that they have going here. The number of returns updates as you type, so when I had only the letter U in there, probably still had 3,339 hits. By the time I got to Ultra, I was down to less than 10. Also, at the bottom of the page, links to the documents that are coming up through your search appear as you type. So once you've put in a word, you can go and look and see if any of those titles seem relevant to your specific technology. So I've been talking for a little bit now, and I'd like to stop at this point and ask if there are any questions on the basics of FDA organization or the resources that exist to find existing information about technologies that may be similar to yours.
Hey, Chris, we don't have any specific questions yet on what you've been covering, so you can continue the presentation.
Okay. Well, thanks. And I just want to encourage people, please do use that dialog box either on the right-hand side of your screen or by hovering over the top of your monitor to post questions as we continue. So let's continue by exploring in a little more detail our case study. We're going to go back to that guidance search that we just looked at using the word ultrasound. What I found in that search was a document popped up that reads Guidance for Industry and Staff, Information for Manufacturers Seeking Marketing Clearance of Diagnostic Ultrasound Systems and Transducers. That seems like something that if I were developing this technology, I would definitely want to pull down, read, maybe memorize, sleep with for a few days, and let it soak through the pillow into my head at night. But there's more than just ultrasound to my technology, and so I also use the search term imaging. And if I am also developing an imaging agent, then perhaps this secondary guidance document, New Contrast Imaging Indication Considerations, for devices and approved drug and biological products is something that I would want to take a look at. I encourage you, as you use that database, to search as broadly as you can for anything you feel may be relevant to your technology development efforts. Searching the term 510K will bring up a lot of documentation and help you understand what might be needed for a specific technology or for the general 510K documentation. So for an ultrasound technology, there actually happens to be on the FDA homepage a section for radiation emitting products. And so they have an A to Z list of regulated products and procedures, and I thought it would be useful to take a look there when developing my case study. When I got to the bottom of the list, I saw that there is indeed an entire hyperlink dedicated to ultrasound imaging. And so as I click through to that, I see that when I get to that page, there is a description, use, benefits, risks, and then about 2/3 of the way down, there's information for industry. Well, that would be me if I'm developing an ultrasound technology. What kind of information does the FDA feel is really important for me to know, so much so that they dedicated time to putting it here on their website? Well, there's information on the regulations and the law that surround the development and maintenance and repair of electronic products and whatnot. And there's also information on policy and how FDA takes those regulations and those laws and implements them. So in addition to the guidance document that we found by searching the guidance database, there's also some other information that FDA feels is really important for me to consider as I'm developing my technology. So let's take a look at that guidance document since we've now found it twice. This is what the cover page of a guidance document is going to look like. It'll tell you when it was issued. If you take a look at this one, notice that it was issued in 2008. We're now in 2016. In 2 more years, this document will come up for periodic review. Every 10 years, the Center for Devices and Radiologic Health takes a look at every guidance document that they have published, and they determine whether or not it still represents their current thinking on the topic in question. If it does, then it gets renewed for another 10 years. If it does not, it gets withdrawn, and potentially a new guidance document is developed to replace it. So, as we take a look at this particular guidance document, we look at the table of contents, and already we're learning something. We're learning that there are different tracks for the ultrasound industry. There are definitions, there are formulas that they expect to see. There is a suggested format and content for how you're going to present that information to the FDA. There's information on how the FDA would like to see you do some statistical analysis of your results of all of your bench testing for your diagnostic ultrasound. As you actually read through the guidance document, there's also this note that pops up that tells you that this isn't a standalone document, but that this guidance refers to a lot of other voluntary standards and other guidance documents. And so you know that this is going to be a good place, a good resource for you to find a lot of the additional information that FDA is going to expect you to know as you continue to develop your technology. Going a little further into the guidance document, they provide a list of product codes for ultrasounds. And thinking about the technology in our case study, it is possible that we might fit one or the other of these 2 technologies that show up in the green box. Well, how are we going to know? How can we determine which is the appropriate one for us and for our technology, if either? Well, one thing you can do is I showed you how the 510K database has a product code field So you can search for something that has this product code, and in the prior slide, those codes were DPW and OBJ. And so I went ahead and I just pulled up one 510K summary from each of those product codes. And if you note over here for the transonic systems technology, it is a simple single product code DPW. And I can take a look at its intended use and determine whether or not this might be an appropriate technology for me to reference for my substantial equivalence. Or if we take a look at the Boston Scientific technology, I see that it has two product codes. It is both ITX and OBJ, OBJ being the term I searched on. And I can take a look at that summary also and determine which is my best fit. So what happens when you take a look at these technologies? What do you actually see in a 510K summary? So this is neither of those two. This is a 510K summary of a slightly fuller technology. But for the anatomy of the 510K summary, I felt like this was a better example. The types of things that you will see on a 510K summary include identification of the predicate device. So if you're considering using technology A as your predicate, what did technology A use as its predicate? Maybe there's something you can learn there. You can see a description of the device. This could help you understand some of the additional potential uses. But in any case, a 510K summary statement will have a statement of the intended use, and that is usually something that comes straight out of the regulations based upon the product code. You will see how the innovator, in this case, compared their technology to their predicate device. You'll see what types of non-clinical testing needed to be performed or that they chose to perform and that FDA accepted. You'll find out what clinical testing, if any, they performed and that FDA accepted for this particular intended use. You may see a number of charts, a number of graphs or tables, and you'll see the manufacturer's conclusion, which is generally a statement that based on their analysis of the data that they've presented, they believe they are substantially equivalent to their predicate device. The final thing that you'll see is a list of references. And I always encourage people to take a look at this, because if you're developing a technology in a field and it's your first time in a product development effort or it's your first time in this particular field or indication, you really want to take a look at the references from these 510Ks, because they often have some of the really key or seminal opinion leaders, key opinion leaders and seminal documentation in the summaries that you will find useful as you continue your development efforts. So we've gone through a lot of this research now, and we're going to revisit our SBIR application, and we're going to consider updating our research plan. Now, we're not likely to actually update the specific aim to perform bench testing, because that is what we're going to do, but in the description of the work that will be accomplished in that specific aim, we're going to expand that a little bit by saying that we're going to test our technology for compliance with the following regulations and standards for Track 1 ultrasound devices, and then we would list a few of them, as suggested in the FDA guidance document, Information for Manufacturers Seeking Market Clearance of Diagnostic Ultrasound Systems and Transducers. And by including just that little bit more information, you can convey to the reviewers that you really do understand how to develop this technology within the U.S. Let's take a look at our commercialization plan also. Rather than simply saying that we're going to submit a 510K for this technology, we're going to expand on that a little bit and say that to access the U.S. market, we'll submit a 510K pre-market notification for our ultrasound device as substantially equivalent to Boston Scientific Corporation's technology that has this 510K number as a ITX, and spell out what that product code is, an OBJ, spell out what that product code is, and perhaps even put in a regulation number, really conveying that you've done some research on this. And when you took a look at that Boston Scientific 510K summary statement, remember before we weren't sure where we were going to be reviewed, which panel we would be reviewed in. Well, it turns out that for this type of technology, this was reviewed by the Center for Devices and Radiologic Health's Division of Cardiovascular Devices. So it was reviewed in the Office of Device Evaluation. So now we've talked about what FDA is asking for, but our technology should be a little bit different, a little bit novel. It should have, some added features or new combination of features. So let's talk about what's different about your technology and how that might confound your development efforts. You want to understand what FDA has or hasn't seen before for a technology that may be similar to yours, or for an indication that you're targeting that there may or may not be a current therapy for. So are you going to be a first-in-class molecule? Are you using a new manufacturing method or new analytical method that FDA isn't comfortable with because they haven't already reviewed a dozen or 100 applications that use that same manufacturing technique or analytical method? If there's no current therapy or diagnostic, for instance, for a disease, it's likely that FDA isn't overly familiar with developing endpoints for clinical trials in that space. You also want to think about things that FDA has seen before. Is there a known adverse event profile, a known risk that you're going to need to proactively address during your development plan? So how can you find out what FDA knows and what they don't know? You could try calling them up and asking, but they'd probably tell you to go back out online and take a look at a couple of the databases that they've put together. So for drugs, you can find out if there's currently an approved risk evaluation and mitigation strategy or post-market monitoring plan for a technology that's similar to yours, either by class of molecule or indication. You can find out what type of adverse events have been reported in the adverse event reporting system. You can look at advisory committee meeting transcripts or listen to the webcast if the recording was successful. The transcripts are always posted. Sometimes the recordings are flawed and can't be posted. But they post every advisory committee meeting online, and if there's a similar technology, listen to not just what FDA brought to the table, looking at their documentation and listening to their commentary, but also what their advisory committee members had to say during that discussion and the questions that they raised. You also want to be aware of any black box warnings for related technologies. So if you're developing an imaging agent, you want to be aware of the black box warnings around gallididium and its impact or its potential risk for people with renal insufficiency. So if you are developing a device, there are similarly databases for devices where you can look up adverse event, failure rates, recall rates, inspection issues, required post-approval studies and surveillance studies, and all of this is, again, available due to the Freedom of Information Act online and is easily accessible to you. So as a final update to the commercialization plan for our hypothetical case study today, you just add in a simple statement. Based upon the totality of regulatory knowledge about ultrasound technology, we do not anticipate specific issues with our technology development. And you could leave it there. But then the reviewers might ask, well, how do you know about the totality of regulatory knowledge? That's a pretty big claim. So you just put something else in there and say, we recognize that we're new to this, so what we're going to do is request a pre-submission meeting with the Center for Devices and Radiologic Health prior to filing our 510K to ensure that we're compliant with all of the regulations, policies, and expectations of the agency, and to ensure that our application will be accepted and reviewed in a first pass, first cycle. And with that, I come to the end of the delivered materials that I have prepared for today, and I open the floor for questions.
Hey, Chris. Yeah, we have several questions that have come in since the last Q&A round. So the first one is, what is the regulatory pathway for combination products?
Always an excellent, excellent question. So the regulatory pathway for combination products is based upon the primary mechanism of action. So if the primary mechanism of action is the device part of a device-drug combination, then it would be regulated in the Center for Devices. If the primary mechanism of action is the drug in a drug-biologic combination, then it would be regulated in the Center for Drugs. So those are always case-by-case studies, and unfortunately, they are, it's an evolving field for FDA. I am hoping to host a future Hangout exactly on the topic of combination products, so please do sign up for our listserv and feel free to come back and join us at that point in time for more discussion.
Good advertisement. So how do you determine what constitutes a predicate device? Is it the operating principles of the device, the intended clinical application, or some other feature?
So the intended use always drives the regulatory path for a device. So your predicate technology must have the same intended use as your proposed technology. Okay.
Next question is, what would be the qualification criteria for the animal rule? Would CDRH be potentially included in future animal rule guidance documentations? Oop.
No, I heard the question. I'm sorry. I'm taking a minute to think about that. That's actually a very good question. Based upon my knowledge and experience with the animal rule, I find it unlikely that the Center for Devices would join in to the animal rule, but far be it from me to stop them from doing anything. The animal rule, for those who aren't familiar with it, is a way to gain market approval without human exposure when human exposure to a therapy would be unethical. So, for instance, to truly test an anthrax vaccine, you would need to both deliver the vaccine and then expose people to anthrax. Not likely to happen. So, the animal rule was developed in order to allow surrogate testing in animal models. I'm sure that there are people out there with much better minds than mine who would have a technology that might, a device technology that might be unethical to test in humans, but I'm not bringing anything to mind right now. If it truly were unethical to test a device in humans, it is possible that the Center for Devices might be willing to work with a sponsor.
Okay, thanks. We have a question that sort of essentially sort of asks how we can anticipate when someone's writing an SBIR proposal and there's a regulatory component in there, how do you sort of anticipate or address the possibility that folks on the review panel might not themselves be familiar with FDA regulations? And so I think we could broaden that question, because we know that the reviewers aren't always familiar with the business proposition or necessarily with the FDA regulations. That's sort of the nature of peer review, and it's kind of just speaking for myself, I'll let Chris comment as well. I think the key thing is to anticipate questions in the same way that you need to convince people that you're a scientific expert on a subject and anticipate potential scientific pitfalls, present alternative plans to convince them that you understand how to navigate the technical challenges. You have to do the same thing on the business and regulatory sides to convince them that you understand the challenges. They're not always going to see eye to eye with you, but you need to present your best case. And I always like to tell applicants you have to basically convince reviewers that you are the expert or you have experts on your team. But Chris, did you have any thoughts on that?
No, I agree completely, and that's the purpose of having gone through our case study today, to go from a very general, non-convincing statement or specific aims and really do the research to convey a much more knowledgeable awareness of the path that your technology is going to need to take. And it certainly isn't an easy thing to do, but very little about being an entrepreneur is easy.
Right. So just there was a follow-up on the combination product question. So you have to propose what you think the path will be and then mention pre-submission plans to verify it. So I guess the question is, what are the steps that one takes if you're not entirely clear on the strategy? But I just, before you answer, Chris, I'll say, just remind people, Chris was planning on presenting another webinar on this topic. So, but go ahead.
But I will answer the questions today also. So if you are not clear about what your primary mechanism of action is for your technology, then you can first try consulting a regulatory professional, right? It's probably worth the money to get an expert's opinion. However, the only person who can truly make or the only group that can truly make a determination of where you will be regulated is the FDA. And so you can submit a request for designation to the FDA's Office of Combination Products. And based upon the way you describe your technology and its intended use, they will make a determination and tell you which center will regulate your technology moving forward if it is submitted as you described it.
Okay.
The problem with that is that it is binding, and it may not be the answer you want. So you want to really make sure that you're clear when you put your submission together that you are proposing the technology as you truly envision it being developed.
Right. So there are a couple of questions also related to combinations or devices for delivering drugs where the drug is either a generic drug, hence it's been approved, or the drug has been, is a subject of a cleared IND, but you have a new device or you're planning on changing the delivery device during the process. What is the regulatory approach that one would take in that case?
I'm sorry, can you repeat the question? I wasn't quite clear on the...
Okay, so you've got a drug, let's say let's say it's a generic drug. It's been, so there's no approval required for the drug,
okay.
But you are developing a delivery technique to deliver the drug to the patient. What is the pathway that one would take, the regulatory pathway one would need to take to study the delivery device?
So, given that the drug is well characterized, it is still likely to be the primary mechanism of action. So it's likely to be a IND filing or an ANDA filing, and it will have a component that will cover the development of the device. In order to gain insight onto the steps required for the development of the device, you can always request a pre-submission meeting with the Center for Devices and Radiologic Health, just focused on the device technology, and there's no limit to the number of pre-submission meetings you can have, although it certainly takes a lot of time and effort for you to both prepare for, conduct, and then assimilate the results of a pre-submission meeting, so you don't want to have them every other week. But you can certainly reach out for any guidance that the agency might have. You can also, for some general feedback on your development plans for devices and for any of the any technology to FDA, you can reach out to a small business group within each of the centers. In the Center for Devices, that's the Division of Industry and Consumer Education, or DICE, at I believe it's at CDRH or at fda.hhs.gov. I'm not really quite sure. We have a prior hangout called First Contact with FDA, and how to reach out to the small business offices in the Center for Biologics, Center for Drugs, and Center for Devices is all included in that hangout also.
Okay. There's another question about regulatory processes for in vitro preclinical tox testing technologies, and I can just answer that and say that if it's used for preclinical drug assessment, it does not have to undergo a regulatory approval. It's outside the regulatory regime if it's not being used to diagnose a condition or treat a disease. I think that's about it. I think we've covered everybody's questions so far. So
then I will put the presentation back to you.
Okay. So just a brief disclaimer to let everybody know that we are presenting these Hangouts for educational purposes. They're not meant to be guidance for a particular product or technology. If you're developing something that falls within the scope of the NHLBI mission, please contact us through our website given here for additional guidance. If you're working outside the scope of our mission, we encourage you to reach out to an appropriate program official in the institute, NIH Institute, relevant to your disease area or organ system, and you can learn more about that at the NHI-SBIR website, sbir.nih.gov. We will be, the slides are available if you email us at either of the following email addresses, NHLBI_SBIR@mail.nih.gov, or you can email Chris directly. She'd be glad to provide you with copies of these slides. We will also be archiving this event and captioning it, and it will be on our NHLBI YouTube channel some point within the next month or so. So look out for that. And again, sign up for our listserv. Gave that URL earlier to get updates on new events and funding opportunities. And with that, thank you very much. Thank you, Chris.